Ladies and gentlemen and ladies, cats and dogs, children of all ages, you know who it is. It is the kid, Unc Just, the pizza freak, resonating at that pizza frequency. Now, what is that pizza frequency? Well, I am glad that you ask. It means I love talking about pizza. I love making pizza, but mostly I love sharing pizza with people that I give a ship about. Yes, literally breaking bread with people that I care about. And I have a sneaking suspicion that you too, my friends, also resonate at said frequency. Huh? Or why else would you be here? And where is here? Well, I am glad that you asked. Today is 8/28/23, 2023. And we are at the free Slow Rise Pizza Zoom webinars. And today we're doing something, the first of three. So we're very excited about this. This is the first of three, Science of Sourdough. Now everybody knows the deal. We are recording. We're recording the video. We're going to use it on YouTube. So if you have fantastic questions about pizza pies and i.e. the science of sourdough, other people can learn and grow from this as well. So thank you all so very much. We're going to what's going to happen is I'm going to bring Noel on. We're going to promote everybody to panelists. If you do not feel comfortable showing your video, that is okay. But we do encourage people to show their video. And if you do have any questions at the end, if you want to raise your hand or put it in the Q&A or put it in the chat, we'll have some time at the end. And we encourage everybody, if you do have a question, let's see your face, and then you can have an interaction with Noel and the rest of the crew. So I'm really excited. Thank you all so very much for coming. Science of Sourdough, Part 1. This is going to be a three-part webinar, and I think we're going to do office hours, and then we'll go back to Science of Sourdough, office hours, back to Science of Sourdough. Mondays, live on Zoom at 5 p.m. with Noel, Mr. Slowrise, and the kid, Unc Just, the pizza freak. And I'm really excited about today. We're going to learn about some sourdough. So without any further ado, I'm going to bring him on. The man, the myth, the legend, Noel, Mr. Slow Rise himself. Noel Slow Rise Pizza, a Brohner. Noel, are you there?
Justin,
Blue Bell. Blue belled you. What'd you think?
Sure did. I mean, I love that bell.
Yeah, me too. Me too. How are things going?
Hot day in Santa Monica. I can't really complain. It's sunny and breezy, a little on the humid side. Not sure why. I guess we're spoiled from last week. It was so beautiful after the
maybe
storm.
so, but we did get a bunch of rain. What's going on? What are we doing?
Well, let's see here. So we are launching our first Science of Sourdough webinar. We're going to be doing it every other week, beginning this week. So it's all new. And so this is our maiden voyage. I'm not sure exactly what's going to happen. I have some notes, 1, 2, 3 pages of notes. I got a lot of attendees here. Looks like we're up to about 65 or so, which is awesome.
We'll see. We'll see we got 30 people right now and participants. So why don't I jump off all the attendees? I'm going to start promoting you to panelists. Once again, if you don't want to show your camera, it's all gravy, because here comes the joke. I'm not wearing any pants. All right, Noel, I'll see you in a little bit.
See you, Justin. Hey, everybody. How's it going there? I'm Noel from Slow Rise Pizza. Welcome to Science of Sourdough. This is our first of a series of, I don't even know how many. I'm planning on doing, I've got 3 scheduled so far every other week. And I've got a lot of people on my wish list that I'm beginning to contact. So let me tell you a little bit about what I'm thinking is going to happen, and then we'll see what actually happens. All right. So let's see. For week 1, I invited one of my oldest, dearest friends. His name is John Porter, and he is the founder. I don't even know if he's still the head brewer at Smog City, because Smog City is so huge now. But back in the day, he was everything, and he and his wife were everything. They wore all hats. So I've known them since before Smog City, and John and I have been making pizza together for the better part of 15 years since I've known him. In fact, if I had to say, he's the guy that dragged me kicking and screaming into the world of pizza. He was going to open a brew pub and wanted somebody to assist with the food. And because I'd been a restaurant and nightclub consultant for most of my life, and also owner of a restaurant and nightclub. I said, let's do either pizza or burgers. I know, let's do pizza. And so we went down the rabbit hole together, and I failed in more ways than one. So cut to 15 years later, and I have a ever-growing business of students and clients who hire me to help them make their pizza dreams come true. And a lot of people have sourdough on the brain. I'm going to say it right now. A lot of people have sourdough on the brain. And so, generally speaking, I have a rule for my clients and students. Let's not start with sourdough, all right? Let's start with like basic straight dough recipes, fresh yeast, active dry, instant dry, whatever you've got. Let's get our recipe, let's get our technique, let's get everything going, and then let's introduce sourdough. And it works very well like that. But there's a lot of people who don't take my advice and they start with sourdough, and they're often in a world of pain. And what I mean by that is that sourdough is its own thing. And I would say that many of us think we understand what's really going on in the depths of our sourdough, but actually many of us probably don't. And so we have good days, we have bad days, we have days in between. But generally speaking, I feel like I don't understand what's going on, and it's hard for me to predict. And so I don't wish that upon my clients or my students, especially my professional clients. And so I steer them away from sourdough as long as I can. And if they insist, fine. I have different methods, different techniques, different recipes, and normally we can get a relatively consistent starter up and running and put it into good use. However, my friend John and I have had many, 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 many conversations about fermentation, about gluten development, about beer making, about flavor, about so many things. And I thought, you know what? If I'm going to do a sourdough Zoom series, why not start with John Porter? Because I feel as if from days of old, artisan bakers all over the world have had a direct relationship with brewers, because back in the day, before Fleischmann's, before any of the big companies started making commercially available yeast, we got our yeast from brewers, from vintners, aka winemakers, from sake makers, from all kinds of people that were also involved in fermentation. In fact, I have tried to make pizza dough with barm, which is, I guess, another word for brewer's yeast. And I've had some good results, bad results, mixed results. I've used yeast from the winemaking process, same thing. I have a client who was really into sake. He was Japanese, and so we worked on using yeast from the sake process. And again, we had mixed results. So anyway, long story short, I thought I would love to have John Porter on to talk about what it is that he gets from fermentation, because I feel like he gets so much more than I get. And so we're going to explore that. But before we talk with John Porter, I want to bring somebody else on. And he's somebody who I met. He's actually a client. He's working on an interesting project that is top secret for now. We'll talk about it in coming weeks. But for now, he's just a microbiologist with a passion for pizza. And so his name is Cameron. I'll be bringing him on shortly when we ring the big blue bell of pizza-ness. Wait for that, Justin. And he's going to give us a very quick kind of crash course in microbiology for bakers. And so this is going to be very quick. This is going to be like under 10 minutes. In 2 weeks, we're going to invite Cameron back as the main guest, as John Porter is this week, and we're going to do a deep dive into that same topic. So that's going to be 2 weeks from now. And then 4 weeks from now, I just booked Stephanie Swan from Modernist. I don't know if any of you have Modernist pizza or Modernist bread or Modernist cuisine, but Stephanie is going to do a history of sourdough with us. So we have a much better understanding of how bakers have been using sourdough throughout the ages, right? How bakers have been using different kinds of yeast throughout the ages, how modern times have affected our process, right? So I think that's really important, because if we're going to go weeks on end exploring, doing deep dives into the science of sourdough with brewers, with scientists, with nutritionists, with bakers, with millers, with farmers, we have to have a decent foundation in microbiology. That would help. A little bit of history, right? A little bit of math, right, Justin? So anyway, that's where I'm hoping to go. So, I actually had a longer introduction written for myself, and I'll try to get back to it. But what I'd actually like to do is not waste any more time, because we're trying to keep this format down to give or take an hour. And so what I'd like to do, Justin, is ask you to please ring your bell, and we'll bring Cameron on.
Right, Cameron, if you're there, let's get that camera on, and I'm going to add you right there, and make sure you're unmuted, my friend. Can we hear you? Great. Yep. Awesome. Okay. Great. Well, I'm going to jump off. It's wonderful to see you, Cam. All right. See you guys in a little bit.
Thank you, Justin. Hello, Cameron.
Hello. How you doing?
I'm doing great. Nice to see your beautiful face. Where are you today, as if I didn't know?
I'm in beautiful San Diego. Also beautiful hot, San Diego. But not too hot.
Beautiful, beautiful. And how's San Diego treating you?
It's good. Yeah, just playing with some microbes, and it's a normal Monday.
Now, how does a microbiologist, an amateur pizza maker, show up on my Zoom? I mean, why should we care about microbiology? Let's start with that. A nice softball question for you.
Yeah, I mean, these microbes that you put in your dough, if you're doing a sourdough, you have a whole collection of microbes in there, a community that work together to leaven your dough, contribute to the flavor, the texture, the lipid browning that you get from that dough, that nice leoparding crust. And most of all, they make the gas, make the CO2 that makes that nice, bubbly, leavened dough that everybody loves.
Now, for those of us who don't know, and that includes me, what the heck is a microbe?
Yeah, so these microbes are just organisms that we can't see with our eyes. So that includes viruses, bacteria, archaea, which most people don't know about. It's a whole branch of life that you won't interact with much. Protozoa, fungi, and algae.
All right. Now, what does all that have to do with pizza?
Yeah, so these microbes are what leaven your dough. They contribute to the texture and the flavor, like I said, but they're ancient. So this is the first thing to keep in mind, is that these microbes aren't new. They showed up 3.5 billion years ago. And if you were to compress the whole cosmic history, so we're starting with the Big Bang and moving until today, if you compress that down into a calendar, from the first of the year to the last of the year, microbes would show up sometime in September, and they would pretty much be the only organisms on Earth until December, when multicellular life showed up. So all plants, animals, everything that and interact with. And humans would show up sometime in the last minute of that year. And during that time, the microbes were making all the oxygen that we breathe. They still make 70% of the oxygen that we breathe on the planet in the oceans. These phototrophic microbes that convert sunlight, like plants, and convert CO2 to oxygen. And you yourself are also microbes. So you're one-to-one human cells to microbial cells. For every human cell in your body, there's a microbe living in and on you.
I mean, that sounds kind of gross.
Yeah. It's gross when you first realize it. You slowly get used to it over time.
Now, is this same grossness also what accounts for like flavor and aroma and color and all that beautiful stuff?
Correct. Yeah. So, for most of human history of interacting and making dough, we were using naturally leavened dough, is what people call it. Now it's called spontaneous fermentation. You mix flour and water, and it makes, something that you can leaven your bread with.
Now, did that happen by accident? I mean, because people didn't have microscopes back then. They didn't have test tubes. They didn't have any of the good stuff we have now, right?
That's a great question. I think we might learn that in a few weeks in the history of sourdough. I don't actually know the question. I don't know the answer to that question.
Got it. But we learned it relatively recently, right? Let's say in the last 200, 300, 400 years.
I think so. Yeah. The Egyptians were using fermentations. The ancient Chinese cultures were using fermentations. As far back as human history goes, there's fermentation, from what I understand.
Now, has bread always been fermented? Do you know that? Has flat bread always been fermented?
I think there's periods in history where it wasn't always fermented.
Got it.
But again, I think a historian would answer that question
Good.
better than I.
Good. So we'll have Stephanie Swan on to answer that question. That'll be excellent.
Correct.
All
Yeah.
right.
I think the key things to know
Yeah.
for John,
Yeah.
before we introduce him, is that there are thousands and millions of species of microbes. So there's, a lot of species that we interact with, but there's even more species of microbes. And there's hundreds of different species found in sourdough and even more strains. So what's the difference between a species and a strain? A species would be like a dog, for example. That's all one species. All the dogs you know are species. And all of the different breeds of dogs would be like a strain. So, for example, the most common yeast we use is Saccharomyces cerevisiae, and that has many strains. Some that are wild and can make some sour flavors, and some that are what we call domesticated yeast, and those don't. They've been breeded in such a way over time by passing them over generations, just like we do dogs, but more inadvertently, to produce more mild flavors. And those yeasts are what convert the sugars to gas that leaven your dough. And then the bacteria are what ferment those sugars into acids. And those are used in all different types of fermentations. So the take-home message is that there's a huge amount of diversity, and there's within each of those species, there's a massive amount of strain diversity. And those microbes work together in communities naturally, and they've existed for a long time before us. And we'll dig into this more deeply and talk about where these microbes come from in your sourdough in particular, and where these domesticated yeasts come from, and a little bit of the history of them next time.
Beautiful. So just so I understand, because I'm learning along with everybody else. So if I buy a package of either fresh compressed yeast or instant dry yeast or active dry yeast, what am I buying?
You're buying Saccharomyces cerevisiae, and in particular about 3 different strains. There's thousands of strains and thousands of different species of yeast, but primarily bakers just use 3 different strains outside of sourdough. So what you can buy on the shelf, and they're all very similar strains. So you're pretty much buying exactly the same organism. They're just manufactured differently. So they either freeze dry them like Han Solo in Star Wars, or they just dry them out, and that's why you need to reactivate them. Like Han Solo, after you get them out of the carbonite, you gotta feed them with some sugar and get them active again.
Got it. Now, when we go to sourdough, is that also yeast with bacteria mixed in? I mean, what's the difference?
Yes, a mature sourdough, so one that's been passed more than 10 to 16 times and kind of no longer smells all funky. Those consist of anywhere between 1 and 3 yeasts and 1 to 10 bacteria, depending on the sourdough. And the yeast is what gives you most of the gas power, so what leavens the dough. And the bacteria produces a lot of that flavor and lowers the pH. And there's a whole dynamic of successions, is what we call them in biology, which just means one replaces the other over time. And they work together as a community. So some of the yeast don't degrade a sugar, but the bacteria does. So it breaks that sugar apart and feeds it to the yeast.
Beautiful. All right. That's a pretty good explanation. I think I'm on the same page. Thank you for that. All right. So unless you have anything you want to add, I'm going to bring you back on for sure to check Jonathan's math and help us translate. But what I think I'm going to do is I'm going to bring John Porter on, and we're going to have a little chit chat with that guy. And then maybe all of us can kind of have a little chit chat together. And I know you're working with James in the background. He's going to be running the chat in case he's got any science-y questions that Justin is incapable of handling, which I doubt that very much, but just in case. So James, hey over there, how are you? Good. So how about this? Justin looks scared.
Yeah.
Justin, I'm gonna ask you to ring that big blue bell, and we'll bring Mr. Porter on.
Thank you, Cameron.
I will see you soon.
Yeah, thank you, Cameron. We'll see you in just a little bit, brother. And I think I'm spotlighted here. I think I am. Okay, so great. I just wanted a little housekeeping. Everybody that wanted to be a panelist from attendee is up. We're going to bring John Porter on in just a moment. Guys, if you do have questions, feel free to hit them in the Q&A, or if you do have questions, you can hit it in the webinar chat. Okay, and then all right, so here's the big blue bell. John Porter, if you're there, let's see. John, there he is. Here he's coming in the spotlight. There he is. Hey, John.
Can you hear me? Hey, guys.
I can. Great. I'm going to bounce
everyone.
off. I'll see you guys in a momo.
Thank you.
Mr. Porter, who are your friends there?
Awesome. Well, I brought my my fermentation products, my couple of beers we make there at Smog City Brewing.
Tell us what you got.
The the classic product pitch. Some of our core IPA, a little Bo Pills, and since we're talking about mixed culture / well, that's what we call our sourdough, if you will. A little white peach saison, one of Noel's favorites, I hear, through the grapevine there. So
always,
yeah,
always,
yeah. And then this other one that I've been drinking while
what's
waiting.
that? Something on tap?
Yeah, a little Czech dark lager.
Czech. Is it yours? Did you make it?
Yeah, of course.
Nice, of course. Oh, nice.
Yeah.
Well,
so
I've known you for a long time. You've always had beer on tap at your house. Has it always been yours?
I mean, when you have unlimited amounts of free beer as the owner of a brewery, that's pretty much what makes it on tap.
I knew you before you had a brewery. I knew you when you were brewing in your front yard.
It's true. It's true. I was. And we were making pizza in the same house in that yard.
We sure were. Thank you for starting me off down this rabbit hole.
Of course.
You're welcome. So I know we have a lot to cover. I'm just going to start by saying that I'm jealous of Brewers because you guys have so many cool toys and so many strains of yeast and access to like labs all over the world, and you can dial in the exact yeast you want and the bacteria and all that stuff. So I'm going to try not to be hostile toward you because I like you and I love your beer. But if you wouldn't mind, I don't even know where to begin, so I'll just shut up and let you talk. Can you break down for us what it is that we can learn from people like you?
Well, I mean, I think it's really interesting when you and I started talking about the science of sourdough, how little knowledge there is behind it, right? And I mean, I've made sourdough bread and maintained a starter, and just as you and probably most of the listeners or the people on this webinar, and that is in complete opposite of what we do as brewers, at least at a brewery our size and sophistication, which has taken us a long time to get to. But our job as brewers is really just to make the inputs for the yeast because they're the ones that really make the beer. You can influence so many things and control so many attributes of what a finished beer is going to be. But, the yeast is really what's doing the fermentation and the finishing of the beer, right? Basically what happens is we're extracting sugar from the starch in malted grains. And then we're cooking that, and adding flavorings like hops, to make, things like IPA. And then we feed that sugar-rich solution to the yeast, which then ferment it into carbon dioxide, alcohol. We like fizzy drinks. We like to get drunk as humans, not just personally. But there's also thousands of fermentation byproducts, namely things like esters, which contribute to the aroma and the complexity of what beer is. And we do this as brewers, basically as pure cultures. At our brewery, we maintain 4 different strains. One for our lager, one for our ale, one for hazy IPA, which is an English strain, and then a Belgian yeast. But we get to choose these strains, and we can basically go to a store or, you know, a yeast lab, and we can, and there's many of them that cater to brewers, and we can just order what we want. And it's like, well, I want these kinds of flavors from my yeast. I want this fermentation performance. I want it to consume X amount of sugar or not. I want to be able to ferment it cold. I want to ferment it hot or somewhere in the middle. And you have these choices, and, you know, experience tells you, you know, what um, kind of what finished product you're going to get. But it's just so funny that bakers, you know, have basically like one option.
Oh, is it funny? Ha ha.
It's like, well, it's the same yeast, and it's also hilarious that it's a Cerevisiae strain, which is, you know,
What's so
Cerveza,
funny about that? That's
Cerevisiae.
all we have. Why is that funny?
I mean, I get it. It's just that, like, you know, brewers and bakers, you know, it was like, okay, we take the stick out with the yeast, and then we put it in our bread, and then we put it back in the beer, or whatever they were doing, you know, back when they called it God is good, you know, before Pasteur and microbiology existed. But yeah, I just think it's so funny. And
How come you're the only one laughing? I don't think it's funny.
I mean,
I'm jealous.
I mean, so much of what we do is to cater to the yeast strains. Just like you would refresh a sourdough starter on a, you know, specific schedule. If
Yeah.
you don't, you know, there could be adverse changes. It gets too sour. You know, I've had ones that just are like, okay, this thing is long gone, doesn't smell good, it's not going to make good bread, right? You know, we do the same with our yeast, but we have very sophisticated tools to be able to gauge the health of the yeast, and you know, and also understand it's what we call fermentation performance, right? So we're every day we're doing what we call slurry counts, right? So the cool thing about beer is that it's not a sticky mass of dough, and when the yeast is done fermenting, it drops to the bottom of the tank. So we can pull a sample and dilute it, and then we can find out its viability, which is like how much is alive, how much is dead. And then we can use that when we reuse that yeast, we can pitch based on that viability. And I know exactly how much to pitch if I want X amount of cells per milliliter in the next fermentation. And the amount of yeast we pitch, whether it's, you know, too much or too little, is going to affect fermentation performance and also affect the yeast health over generations. And with these tools, I think our house ale and our lager strain, we're buying maybe once a year, and we're going between 60 and 80 generations, which, I mean, we're reusing it, and each generation could pitch multiple fermentations. So we're using these things 100, 150 times before we have to go back to the lab to buy another one. Again, like, I don't know why we just have so much control, and I don't know how that translates to bread making, but it seems like something that's worthwhile, you know, investigating, right?
That's why we're doing this, because it's like all these people have one key to one door of the castle, and in that castle is all the secrets of yeast and sourdough microbiology. But even the microbiologists don't know everything. The historians know some things, the farmers know others, the millers know others, right? The brewers know others. So I'm trying to get people together. And I mean, I look like I'm joking because I have a shit-eating grin on my face, but I really want to understand because I'm jealous. And also, if you guys can make such good beer with what, 4 different strains of yeast, imagine what
Yeah,
bakers could make if we had access to even two.
I know. I mean, there are hundreds out there. I mean, I can
I
just, it's a
know.
huge list. And you're like, what do you
I
want your
know.
beer to taste like? What are
Well,
you going to do?
I know. I mean, I
You
have this
know,
fantasy that like you would use one kind of yeast for like a pan pizza, right? Maybe another
yeah.
kind of yeast for like a thin crust pizza that like goes like really long on fermentation in a cold environment, you know? Maybe another kind of yeast for like a bread that's like an overnight room temperature rise, right? Never touches a refrigerator. Right. And then,
Yeah.
like, I mean, also, like, I've worked with a lot of different pre-ferments, is what we call them. And so, like, it's one thing to use what we call a straight dough, which is where you add a fresh or an instant yeast to a dough. But it's another thing to ferment your pre-ferment with yeast and then add it to straight dough or add it to sourdough, and then you get
Mm.
some real subtlety. I've talked about it before in my advanced one class, but I remember when I was first getting started and picking up one of the Chad Robertson books. If for people who don't know, he's the tartine baker, and he had this recipe for a baguette which had one kind of levain or sourdough and two kinds of pouliche, right? And I remember thinking, what the hell? And I tried it. And it never came out exactly the way I wanted it to, because timing each pouliche, like a 4-hour and a 16-hour, and your levain perfectly so that they all like come and sing to the angels at the same time was like impossible. So I got 2 out of 3 both times. But I got to say, 2 out of 3 is pretty awesome. Hold on for 1 second. It appears that my computer is not charging. What is that?
I just got a notification that the Science of Sourdough webinar is happening. Well, so the one thing I will say that we do really have in common, right? So like, because I'm sure bakers are like, or people making pizza are like, well, what does pure culture and all this control that you have to do with any of that? You know, what I do at home or at my business. And the other thing is that like we have a separate facility where we produce what we call sour beer. You guys have probably heard the term. Mixed culture, wild is like, we don't have great terminology to talk about these beers. But we call it a sour program or the wood cellar. We are using mixed cultures. Some of them start out as pure culture yeast that we introduce into either large wooden, call them barrels, fooders, storage tanks, and they develop. It's funny, we when we have sourdough or sour beer, we call it an infection. It's not a very charming word, but they'll develop a range of microbes. Sometimes up to 15 or 20 different microbes in there. Sometimes with a beer like this, we'll start out with pure culture, or maybe it's got some wild yeast in it, and then we're processing whole white peaches. We don't wash them. We just kind of crush them and chuck them in, and we get, different fermentation byproducts. So, we're handling, these mixed cultures, which largely the things like lactobacillus. I don't know if you guys mentioned that earlier. That's what makes lactic acid, makes sourdough sour, makes sour beer sour or tart, whatever. Those are largely known in my industry as a beer spoilage organism. And so,
Sorry, John.
that's okay.
Noel has to jump off for 1 second, guys. He seems to be having some issues with his computer. So, Cameron, I'm gonna bring you in and ask you to unmute, because and you guys can continue this discussion. All right, guys, I'm still here. If you have some questions, throw them in the chat. If not, take it away, gentlemen. Thank you very much.
Awesome. Thanks. Awesome. Yeah, so it's kind of like what we were talking about the other day, Cameron. It's like, mixed culture sour beer, they find all kinds of stuff in there, right?
Yeah, and there's a whole community of organisms that are coexisting with each other in a sourdough. And I imagine they're doing a similar process in your sour beer.
Yeah, and we have ways of controlling them, just like bakers can control how much acidity they have. If you want that leavening power, you need yeast more than you need lactobacillus or, the bacteria. It's the same thing for us. We don't want the beer to be too sour. We're able to manipulate the inputs. Lucky for us, we use hops, and hops are somewhat antimicrobial. So we can sort of favor the yeast over the lactobacillus or lactic acid bacteria. But yeah, it's a lot of it's making sour beer. I always say it's more like winemaking, because there's a lot of intuition and things you learn over time, which was the same in my experience with sourdough cultures. It was like, oh, if I leave it out for too long, it's not good. If I leave it in my fridge and I don't feed it for 5 days, it's not, it's like things you learn over time. Which again, I don't know. I mean, I guess if you are really into sourdough, there's a lot of information on the internet, but I don't know if there's like a go-to place where you're like, here's all the do's and don'ts, because your culture is your culture, right?
Yeah.
That's
Yeah.
the difference.
I think the difference too, I mean, from a microbiology standpoint, is that you know exactly which strains you're putting in for sour beer, right? So
Mhm.
you kind of know their characteristics, what they should be growing at, you even know their viability. And what that means is essentially how many cells there are per milliliter that you're putting in, right? Um,
Correct.
versus when somebody makes a sourdough, unless you at home have access to sequencing technology and a degree in molecular biology, you're not going to really know which organisms are in your sourdough. And it could be any, I mean, there's hundreds of different yeasts, specifically at the strain level too, and hundreds of different species of bacteria that could be showing up, even though some are more dominant than others across the sourdoughs that are made. Um, but it could behave totally differently. And because of that, it's almost like you have a zoo of organisms with a
Yeah.
sourdough, and you just removed all the walls in the zoo. And your way of like trying to keep the zoo afloat is by just like, okay, I want the penguins to go up. I'm just going to try and make it colder, but you don't really know there's penguins in there. And it turns out it's
Uh,
polar bears, and then they eat all of your cats or something. You know what I
yeah.
mean? Versus what you
I
have,
love this analogy.
you kind of you have the zoo and you know which animals are there. And I
Yeah.
think you have the ability to manipulate it in that way and feed. Okay, I want my lactobacillus. Make this one a little bit more sour, so I'm going to add a little bit
Yeah.
more.
Yeah.
You know,
Keep it hot.
yeah,
Yeah. Yeah, absolutely. And even on like our wild side or in our sour cellar, you might see things like wild yeast or a wild beer on a beer label or Brettanomyces. That's like, a thing that
mm,
people who are really into it put on there. But again, like we're buying these from a lab. We can get
yeah,
pure cultures. We can get multiple strains of Brettanomyces. Like I know
hmm,
exactly what their fermentation performance is going to be like. I know what they contribute if I bottle condition, if I do a
hmm,
primary fermentation. Like
yeah,
they're wild, but they're domesticated.
yeah,
It's like having a pet wolf. It's a wolf, but it was born in captivity or, raised by somebody who it's that kind of thing. I mean, there are people who go out and do wild, air
hmm,
harvests, and there's a company
hmm,
called Bootleg Biology where they send you little kits and you can, put wort, in a container and put it in a tree in your backyard or go into the forest. And like, there are definitely ways of harvesting wild yeast, and there's some really fantastic,
hmm.
spontaneous breweries that embrace that. Um, but largely, those processes take a lot longer, and they're very expensive, and so they tend to be like more niche and harder to find. So,
So in those cases, you're isolating the microbes from an environment and using that isolated
yeah.
microbe. So maybe we could explain that to people. What's the difference between enrichment like a sourdough, which has a whole community, and what's an isolate? What does it mean to isolate a microbe? For beer
Well, I
or in
think
general?
Well, I think you there are two things you want to accomplish. You want it to but you want yeast to ferment, you want it to
Hmm.
consume the sugars and make CO2 just like I do, and you want it
Hmm.
to taste good,
Yeah.
right? And so you go out into an environment and you give it the substrate that you're going to be using because if you get a yeast or a bacteria and they don't do anything, and they're in there,
Yeah.
then it's totally pointless, right? Or they only go halfway,
Mm.
and you're like, oh, well, I can't make bread with this, or like, I can't make beer with this because it's only went, for us, attenuation is how, the level of sugars it can consume. The funny thing about maltose, um, which comes from malt, which is made by malting, sprouting malting grains, is that it doesn't exist in nature
Mm.
very often. Bacteria can break down starch and create maltose, as a sugar. But to find things that will consume maltose and actually taste good, you're really narrowing that window. Whereas, things that can consume other simple sugars, like fruit sugars, like, they say the first fermented beverage was mead because yeast in the air got onto honey that was in a hive that got wet, and somebody came along and drank it and got drunk and said, this is the best thing ever. But they're not going to say that about beer because beer is made from malt and maltose, and there's not a lot of things like the yeast that you find on grapes, and things like blueberries. I knew a guy who worked at a bakery, and he used to scrape the white stuff off of blueberries, and that was how he made their sourdough culture. And those things for me wouldn't work because they don't ferment maltose, which is the predominant sugar in beer. But I mean, you can get it from a lot of places, right?
Yeah.
I mean, where do sourdough bakers get their yeast from? I mean, are there people going out doing wild capture and stuff?
So sourdough is spontaneous. So all of the microbes, unless you maybe buy a dried sourdough on eBay or something like that, which in that case, we don't even know which organisms might be alive in that,
or like the San Francisco pier,
right?
The bread bowl and the little packet. Yeah.
You go to your local bakery, they normally give you a sourdough if you ask for it, if they're making it. But in that case, yes, it comes from, there's a few studies on this, and we'll talk about it in 2 weeks in depth. But most of the microbes come from the flour and from the person themselves. So
I heard that. I've heard about the hands.
Right. Yeah. So the microbes, a lot of them that are in sourdough can be found on your hands. They can also be found in the flour. And so it takes careful backtracking to figure out, what's the percentage that come from each one. But the microbes that are in flour, a lot of them come from places that people wouldn't expect. So just because it's coming from flour, it doesn't mean that it was necessarily part of something you'd expect. So a lot of them come from insects.
I was going to say from like the farm. Does it come from the farm, or does it come from the people who grind it into flour?
It comes from the soil. It comes from the insects. It comes from the microbes that live on the outside, right? These insects bring the microbes from plant to plant. So Saccharomyces cerevisiae is commonly found in flies. Same thing with lactobacillus, or now it's called Fructolactobacillus sanfranciscans, which is a common bacteria found in sourdough that makes the acid and makes it sour. Those are found on insects all the time. So people think often that they might have even originated from there. It's hard to backtrack fully where a microbe's original niche was, or where they best existed. But they come from all over the place. They get ground up in the flour. There's even microbes that live inside the plant cells itself. So all these microbes get ground up into this powder, right? And along with the insects, whatever else, the soil that gets contaminated on there. So that's what populates your sourdough, and why there's so many different types of sourdoughs.
And bacteria is so small, like it doesn't matter. You grind flour, you're not hurting them.
So exactly.
They're so insanely small.
You could fit dozens of them. They could dance on a human hair. That's how small they are.
Yeah,
very, very, very
amazing.
Small.
Yes, that's why our beer spoilage organisms. So we have two separate facilities, because they're so small. They're very difficult to eradicate. So our difference between what we call clean beer and sour beer. We actually have a parking lot in between, and all of the sour beer is produced in a facility that's separate, to make sure that there's no cross contamination.
Yeah, they love to grow.
Yeah, they do. They proliferate, but like I said, we have tools to sort of keep them down, which is good. I don't know if there's anything in baking where you can control the amount of like lactobacillus versus yeast. Like I imagine that population is constantly in flux, right? If you're doing your feedings, if you want it to be more sour, like, hey, I want to make it like San Francisco style, which I swear they just pour lactic acid in. Or if you want it to just, to have like more lift and be more predictable, and there's got to be ways of controlling that.
Yeah, this is where the bakers like Noel have the experimental experience more than, there's so much conflicting information about this, even in the science literature, about, which mechanisms control things like this. And I think bakers might argue about it too. Noel can correct me if I'm wrong on that. Is that true and all? Do bakers argue about, how to manipulate their sourdough?
Oh, we argue, but not with facts. We hurl insults. Where people say, "No, you don't know. You're only 1% Italian." I'm like, I'm 2% actually.
Doing it wrong.
Listen,
That's what they say.
well, here's the thing. I don't think. I mean, I'm back now. By the way, I had to replace my charger. It died. But while I was replacing my charger, I was listening to you guys, and I thought what you were talking about was pretty fantastic. The level of conversation that I just witnessed between you two has never happened between two artisan bakers. Not really. Not bakers that I've met. And I've met a lot of bakers. And so I feel like that's a conversation for the future, but I don't know what it's going to take to propel us to the point where we take an interest, because I feel like we're a little stuck in our ways, right? And you use either active dry or instant dry or fresh. You either do a straight dough or a dough with 30% poolish or 20% biga or 40% pate fermenté or 50% sourdough or 2% sourdough, whatever it is. Stiff sourdough, liquid sourdough, right? This is like, it's time and temp and amounts. That's all the parameters we're really dealing with. And I feel like I don't know what it's going to take to jumpstart the community, because either we're steeped in tradition or we don't know anything and we're just like throwing dough at a wall and trying to see what will stick. And that includes me. When I first started baking, I came out of French Culinary Institute armed like with the wisdom of a sophomore in college, thinking, all right, I'm going to make awesome pizza. I'm going to make sourdough with 40% leaven. And I made it and it was awesome. And then the next day I took it out of the fridge and it was so sour. But I grew up with San Francisco sourdough from Boudin. And so I was like, this is great. This tastes like Boudin, right? And so, it's not very often that conversations change, I feel, in the realm of pizza and baking. But I feel like there's a new generation of pizza makers. And the funny thing that I'm seeing right now is that a lot of the professionals, and by professionals, I mean professional bakers, but also like companies, they're looking to home bakers for what they're doing. Right. And you can see it like at the Pizza Expo. You can see it in the marketing of companies that go to the Pizza Expo, which used to be a 100% professional event. But now some of these companies are featuring home bakers because they have more influence at times than some of the superstars professionals. Right. Because the thing is, they can afford to do a Chad Robertson style pizza modeled after his baguette, because they only have to make one or two good pizzas in a night, not 1,000 or 2,000 like some of the pizzerias I work with, right? Not 60 or 80,000 like some of the chains that I work with, right? So what's interesting is that what I've seen in the last, let's say, 4 or 5 years is that there's a whole new model for baking, and it's really about throwing out tradition and trying new stuff, right? Or it's what's actually new is not new. They're actually bringing back old stuff, right? When you talk about slow food, right? When you talk about traditions from hundreds or even thousands of years ago, when you talk about like fermentation, that's what I'm talking about. But what you guys are doing is you're taking like these old traditions, you're taking slow food, but you're adding science to it, John, and that's something that separates professional brewers from bakers. And I'm pretty sure that some of the big bakeries in this country have microbiologists on staff, and probably they're digging deeper into yeast and bacteria and microbes, and they're doing stuff that we're not doing. But why shouldn't we have access to some of the same technology that you guys have, right? What's it going to take? So we're here to disrupt. We're here to make some noise. We're here to start screaming, and we're going to bring professionals on and see if we can't, enlist some people with who maybe have the ear of some of these big companies or somebody, so that we have access to think about home brewers, John. Don't home brewers have access to a lot of these same strains?
Yes, absolutely. You can get them just the same exact thing, liquid cultures.
Right.
And the cool thing is that dry yeast has come a really long way. Like, it used to be like my mother-in-law was brewing beer on base in the, early 70s, and she was right next to the baking aisle was the malt extract, and she was buying the same Fleischmann's, whatever. It wasn't any different. Um, and dry yeast was a joke when I first started homebrewing 20 plus years ago. And now most of the strains, like the technology has come a long way, and you can get, people have a dozen strains you can use, and you just chuck them in. Now you don't even have to hydrate them or check them; they just chuck them in. Like, that's a selling point, and you can pick all these different strains. My argument is that, like, the first fermentation isn't the best one. It's usually like 2 or 3 generations, to reuse, to kind of get the yeast used to the environment that you give it, that you need it to function in. But it's still really versatile, and a good backup plan to keep. It's got a great shelf life, and you just keep it around.
Yeah.
So, but again, that's pure culture, right? Pure
Let's go
culture
back to
is
that, because I have some questions about that, because I know there's some parallels, but I'm not sure I understand what they are. So were we trying to make the connection that pure culture is like our yeast, and like sour culture that you use for your sour beer program is more like natural fermentation or sourdough? Is that what we were saying?
yeah.
And
Eh,
and a quick follow-up question: that, and what is the difference in like the realm, the library of flavors, aromas, all the byproducts that you get from fermenting, a pure form and a sour form?
I mean, the answer is always going to be it depends on the strain, because there are so many. But pure culture tends to be predictable, which is why people do it. And beer making is an extraordinarily cleaning intensive process, because we are always maintaining the sanitation of our equipment. And so yeast handling, and for the most part, especially at our facility, is done without any person's hand. In fact, it never even touches the air. They're in sealed tanks. They go through hyper clean hoses and vessels if we're going to move them from one place to another. And so, if you started with a pure culture at home and you wanted to maintain that, without all those protocols in place, I don't know how you would not become like sourdough or some sort of mixed culture.
Yeah,
So,
that's a good point. Let me switch over to Cameron real quick, because Cameron and I, when we spent a few days together in San Diego, we talked exactly about that. Cameron, are we going to in the future get into some conversations with scientists who can explain to us what happens when a culture gets adulterated, and what parts of it kind of remain the same and intact, and what parts don't? Do we even have that kind of understanding?
Yeah, and there's some things like, for example, the sourness and the disagreements of, whether or not you make your starter dry. Does it make it more or less sour, right? That actually depends on the lactobacillus strains that you have in your starter. So, for example, it's generally thought that making it dry can make the yeast grow better and can make it less sour that way. But if you have Lactobacillus plantarum or Lactobacillus systems, uh, it actually does the opposite. Those microbes will take over in a dry starter when present. So not knowing whether or not those are in there, it's really hard to figure out. And of course, you're just going to default to which starter, your starter at home. And there's a lot of people who have spent their lives studying this, and we can get them on here and talk about all of these nuances, right? And I think we, as the scientists, also have a lot to learn from the experience, to ask the right questions. If you're not asking the right questions in science, then you're not really doing anything worthwhile. You can spend a lot of money in science doing something not worthwhile if you don't ask the right question.
Yeah, yeah. Well,
yeah.
I feel like in a lot of ways, bakers aren't asking the right questions. Right. And I'm not trying to dig on bakers, but because we have our hands full with a lot of other things, but there's at least in my world, you have sourdough bakers that think they have the market cornered on flavor and aroma and nuance, right? And then you have people who are using commercial yeast, and you have to go to incredible extremes to coax, certain flavors out of a yeasted dough, but it's very limited,
Yeah. And to the point that John made, I think uh, beer making is, a lot more fine tuned. If you get a contaminant in a sourdough, because you're working with a community that's pretty resilient to an invader, an invasive species in a community is going to have a hard time taking hold unless you leave it for a long time. You get some mold in there or something. But that's why you can revive sourdough so well. It's a very resilient community with a lot of redundancy in how it functions. And we've been able to, bakers have been able to rely on that versus brewers. I think probably, my guess is that they had to switch to this stuff earlier by necessity, because just completely working with wild fermentations is probably much harder than it is in baking. That's my intuition on it. Even though I'm a novice in those two.
Yeah, I think the problem with pure culture is that there are breweries, they started to do it, right after Pasteur started isolating, and they were doing it at the Carlsberg Brewery, and that became the standard. And there are still people doing spontaneous fermentations in Belgium. There's areas where they still make lambic, but different flavors. Like that's the thing. And I think the biggest difference between pure culture, or actually to comment on the sort of longevity factor, is that pure culture yeasts they live and they die quickly. Like we're reusing them every 5 or 6 days, or else we have to harvest and store them as close to freezing as possible. And we only get another 2 to 5 days before we have to reuse them. Whereas I have a 3,000 liter oak tank, and I can leave it there for 2 months, and then I can add more work to it and it'll ferment, and then I can blend it and put it into beers like this. I can do a Brettanomyces starter and then use it 30 days later. So there's more flexibility there, for sure. But you're right. I mean, out of necessity, these pure cultures, they're real finicky. And yeah.
We have a comment and a question coming from the crowd. Justin, do we have anybody
Katie,
who wants to be promoted?
Katie, did you have your hand raised? Did you have a question, maybe?
Hey guys, yeah, just a quick question. Appreciate this conversation so much. How can I find out the microbial contents of my sourdough starter? Like, is there a service or a person, or maybe Cameron, can I send it to you? Can you sequence my starter for me? I need to know. This is like, you've done this to me. Now I can see I need to study microbiology in addition to sourdough. Great.
Well, we might do something in the future to try and do this, but if you send me a message, I think my email is probably somewhere around here, but I can send it to you. We can talk about it.
Bless you.
Yeah.
Kate, were you in our office hours last Monday?
Yes.
Yes, I was.
From San Pedro?
Yes, San Pedro sourdough. And I
love it.
Yeah, and I love Smog City, by the way. Hey, neighbor.
Awesome. Welcome back.
Thank you. And I did all of your 3 suggestions for my pizza dough. Wow. Thank you so much.
Good. Well, that's the beautiful thing about what we do, because I'm not a scientist, but I understand what happens in fermentation, and so it's really just time and temperature. So I hear things that scream at me when people say what they're doing, and I just whisper back at people, try this, try that, don't do this. And it seems to work, so I'm going to keep doing it. But thank you for your question. And Cameron, you keep us posted and let us know what our options are moving forward. I'm sure that things are just getting moving, and maybe in the future there'll be a service where people can have their well, I mean, hell, you can swab your tongue and send it in, and you can get your DNA sequence. So how hard could a starter be, right?
Thank you so much, Kate.
Beautiful.
There are definitely places that will take your beer if you're making fermentations, and they will genetically sequence everything. I'll tell you exactly what's in it.
Really? Is it expensive?
I don't know. I don't. I mean, it's does it taste good? Does it work? Let's go with that.
I don't know. Let me read a comment, not a question, but a comment that we got, and see. I want to see your reaction. All right. Here it is. A lot of brewers' yeasts are selected for flavors that could easily evaporate and be lost in bread baking. Could that be why we don't see more yeast option in bread? Many bread parameters are focused on time and temp.
I mean, it's plausible, but a lot of beers are pasteurized, and they still maintain things like esters and fermentation byproducts. And that's either flash pasteurization or tunnel pasteurization. those are pretty hot. I mean, they're not 300, 400, however many degrees. Um, but not everything is lost, you know? Um, I don't know. I feel like you'd have to try it,
Yeah, no,
you know?
that's a
Yeah.
pretty good
And
answer.
so like, we don't boil it after we ferment it, so but pasteurization is pretty rough, and
Yeah.
you can still get the ester profile of a Bud Light, even though it's subtle and weird. Um, it's there.
Yeah, I think it's not just that. It's like you said, you have these microbes isolated, right? So they're picked out of their community, and you can control their concentrations. So in somebody who's making sourdough, which I think is the most distinct flavor from your just Saccharomyces cerevisiae bread yeast that they're typically using, you don't have any control of the microbial density in that sourdough, really, outside of external parameters of, like we were talking about, make it dry or make it higher hydration to try and control those things. But you could make your lactobacillus way higher if you want to increase the sourness, like we were talking about. And that's even after you pasteurize it, for example. And a lot of these molecules that have the flavor, they don't denature it at that temperature. Not all of them. So
I would
and
argue that
denature means fall apart.
yeah,
Justin, keep
right.
me honest.
I would argue that a lot of these things we're talking about is subtlety.
Yeah,
Things like esters in beer, and like you need almost a trained palate to identify what they actually are. And so then the same case might be said about, those flavor compounds in sourdough bread. I don't
yeah,
know. I mean, I've certainly smelled sourdough starters where I'm like, whoa, that thing is so fruity. Why is it so fruity? And
yeah.
it's,
Well,
or you get that blast of alcohol and you're like, oh yeah, definitely fermentation. It's
yeah.
boozy.
I think when we talked about it last week, you said it was the character of 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 the beer. I think that's kind of like a good analogy for it, is that you have the canvas and the materials, right? Which is like your sugars in your flour or your dough, or in what you're going to ferment for the beer. But what gives it the character are these microbes that convert these sugars into all of the different metabolites, or these small chemicals that create the sensory impact on the end product. It's the character. So exactly, it's less in your face unless so you had the lactic acid directly in with maybe with the sourdough before doing San
Right.
Francisco. I don't know. It's pretty sour.
Yeah.
2 weeks
Yeah.
maybe
Well,
fermented.
I mean, I think most people are trying to get good fermentation. I think, I mean, my experience in trying to make sourdough bread was really about trying to get to master the process in a way where I was learning also just how to make bread. I wasn't like already good at making bread. And so things like timing and, how I'm mixing and all of those parameters that go along with the process
Mhm.
were there. I didn't have time to like evaluate what my culture was actually contributing because I didn't know, I mean,
Yeah,
I feel like, evaluating what something is bringing to the table or not is a very advanced um, undertaking.
yeah,
So, yeah,
cool.
Awesome, gentlemen. My head hurts just listening to you guys. I'm
perfect. That
serious.
was my goal.
This has been fantastic. Unfortunately, our fearless leader seems to be having not only energy computer problems, but also having some internet problems. So,
Uh,
guys, we had one last question from Dominic.
mhm.
How would you incorporate beer into sourdough pizza dough?
I mean,
Pour it in there. Use it instead of water. I don't know. Why not?
well, I mean, you're using it for flavor, right?
Yeah, yeah.
Why else would you use it instead of water or, like whatever, it's part of it, right?
Or
You're not going to use it for carbonation, right?
right. And
We're not like beer battering things. So
No,
for me, you would pick it based on what flavor contributions you want it to bring to the table. I would stay away from hops, anything hoppy. I mean, you can, but what a lot of people don't know about hoppy beer is that the bitterness comes from boiling or cooking hops, and the change happens at higher temperatures. And there's a lot more hop character in here that hasn't had that change invoked. And when you cook it and when you bake with it, it makes it happen. So you think you're gonna get this flavor, and then you just get tons of bitterness. So that's like sort of like a little word to the wise.
Awesome.
But I mean, otherwise,
awesome. Well,
beer away.
Yeah, well, this has been fantastic. Do you guys want to say thank you to all of our attendees and participants? Why don't we start with you, John Porter, and then jump over to you, Cam, and then we'll say hey to James in the background. James, thanks. I know we didn't have a lot of, I'm going to spotlight you real quick here. James has been in the background in case. James, do you want to say hi?
Hey, everybody.
Yeah. So I believe next week or the next sourdough class, we'll have Cameron and James here, and we're going to be deep diving even more into the science of sourdough. So, John, why don't you say your goodbyes, and I'll take everybody off. So.
Great. Yeah, I just want to thank everybody for coming and hearing what a brewer has to say about yeast and sourdough, and hopefully you learned something. And I really want to try that sourdough pizza dough with beer in it. So you look me up.
And everybody, you can find John down at Smog City Brewery. So thank you so much, John.
Yep.
And Cameron, you want to say a few words, and then we'll.
Yeah, thanks for coming. Think of some good questions for 2 weeks from now, so I can prepare some good answers and do my research ahead of time. And I'll see you guys in 2 weeks. Thanks for coming.
Great. Thank you so much, Cam. Really appreciate it, bud. And James, thank you for being in the background. We'll see you in a few weeks, okay? Guys, at 6:06, the first round of Science of Sourdough, other than Noel's internet and his computer glitching out and all that stuff, I think it was pretty good. There was a lot of information there, and we're going to keep this up. Next week at 5 p.m., same bat time, same bat channel, we will have our regularly scheduled office hours with me and the maestro, and then the week after that, we'll be back on the Science of Sourdough. So, the deal. I want to say thank you. What do I say? I said, remember to be long with one another, the opposite of being short, because we're all in this together. We don't get into the politics of pepperoni and pineapple. We say pizza always, and always pizza. Pieces and slices, everybody, with love. Thank you, everybody. We'll be seeing you. Bye. And now I start to unparticipate everyone. And you can sit here with me while I unparticipate, or you can just turn yours off too. All right, guys. Thank you so much. Chef Serge, I see you. Ezra, I see some of our friends. It's great to see everybody. Sorry Noel isn't here to hang out. He his computer's not working, his internet's not working, but it's great to see you guys. So thanks so much, everybody. All right. We'll see everybody. Sometime soon. See you next week with love. All right. I'm out.